dexamethasone, a commonly used medication in equine practice, on the antibody response of horses to vaccination. Dexamethasone is a type of glucocorticosteroid well known for its anti-inflammatory and immunosuppressive properties. It is frequently administered to horses to manage various health issues such as asthma, allergic reactions, inflammatory bowel disease, and even certain types of cancer like lymphoma. While the therapeutic benefits of dexamethasone are well established, there has been limited understanding of how this medication might affect a horse's ability to develop protective antibodies after vaccination. Some earlier research has touched upon this topic. For instance, one study suggested that inhalant fluticasone, another type of corticosteroid, does not seem to interfere with the immune response to vaccines in horses. Another study indicated that a single dose of dexamethasone could lead to a decrease in the production of certain immune cells, specifically CD4 plus T cells, and reduce the levels of inflammatory signaling molecules in healthy horses. Furthermore, it has been observed that repeated intramuscular administration of dexamethasone to horses following vaccination against bovine diseases resulted in lower levels of specific types of immunoglobulin G, although the overall levels of immunoglobulin G in the blood remained similar to those of vaccinated horses that did not receive dexamethasone. Interestingly, the effect of immunosuppressants administered around the time of vaccination is also a subject of ongoing investigation in human medicine. Several studies examining the impact of corticosteroids given orally or through inhalation on the response to different human vaccines, particularly inactivated influenza and COVID-19 vaccines, have not consistently shown a reduction in the immune response in treated individuals. Currently, the Centers for Disease Control and Prevention have not issued specific recommendations regarding the use of corticosteroids before or after vaccinations in humans, although they suggest reducing their use in such situations whenever feasible. They suggest that the effects of dexamethasone on the equine immune system are complex and not fully understood. Different dosages and administration schedules of dexamethasone or other corticosteroids could have varying impacts on antibody responses in horses, and these have not been adequately studied. Furthermore, this study primarily focused on the antibody-mediated immune response and did not investigate how dexamethasone might affect the quality of the cell-mediated immune response and the activation of T cells in response to the vaccine. Additionally, all the horses in the study had been previously vaccinated, meaning they had an existing immune memory. It is known that secondary immune responses after repeat exposure to an antigen are often stronger and involve memory cells. Immunologically naive horses might respond differently to corticosteroids given at the time of vaccination.